Good morning. It's my pleasure to start this webinar on big data in immunology and specifically in the era of COVID-19. These webinars are organized by the French network FAI²R on autoimmune and autoinflammatory diseases. My name is Vassili SOUMELIS. I'm a professor of immunology and clinical hematologist in Paris at the Hôpital Saint-Louis and director of the human immunology department at the INSERM. So I start with this very classical view of big data by numbers, and we can all be very impressed by the volume and the velocity at which these data are being generated and represented here, both in according to the volume, so gigabytes, terabytes, petabytes, exabytes, and so on, and also with images related to the volume in terms of stacks of DVD and distant scenes and how many buildings and so on. So this is a computer scientist or a data scientist view of big data. Here, and this was at the origin of the famous 3V to define big data: volume, velocity, and variety. I want to introduce a different view, which is a medical doctor's view and biologist view that is related to the information contained in the data rather than their volume. And here in the middle you see basically the information represented in terms of biological insight, how much we can learn from big data in terms of the function of the biological system, its structure, molecular networks, and so on, an organization. And on the right, how much we can learn from big data in terms of clinical impact to improve prognosis, diagnosis, and treatment of human disease. And we'll go through some aspects that are very important and can really impact the quality of the information, the conclusions drawn from big data. So a very important aspect of big data is the data life cycle from the source to the final biological or clinical message. So when we get data, when we try to learn from data, basically the information contained in the data depend on parameters that can be anywhere in this sequence of events, and the information can be influenced or distorted by parameters that can be in any of those steps. So from the left we start with collection, meaning starting with a patient or a model organism, and you can already have important aspects there, and then clinical sampling, and then preparation of the sample in terms of cell isolation, DNA extraction, and so on, sequencing or any other data generation technology. Bioinformatics is meant for data analysis using different methods, and then interpretation of the analyzed, which leads to a biological or clinical message, meaning the conclusion. So why is it so important to control and streamline that process? Well, it's because at each one of those steps there is the possibility of bias, and it's not only a possibility, basically there are biases that are intrinsically contained in each one of those steps, and the fact that we can control the process and have a good knowledge of what happened at each one of those steps puts us in the best possible position to know about the existence of those biases, to control them, meaning preventing them if possible, and to account for the biases in any computational bioinformatics, biostatistical analysis. So, is it always possible to control all this process? Well, unfortunately not. Sometimes we obtain data without getting information on the initial steps from patients to samples to technologies and to technical parameters having led to the generation of those data. And in that case, the biases that can be associated to those early initial steps cannot be assessed, estimated, and sometimes are not even known. And so this is the case when we try to exploit real life or real world big data, because most of the time when we have access to the data, we cannot reconstruct the upstream events of this data life cycle. And so these big real life big data and any biological data that are publicly available as resources to a scientific community have their pros and cons. So the strong sides are that sometimes they are very large in terms of data types and volume, so with a big potential to learn from them. There is a lower cost, at least for the people who exploit them once they are generated. The cost can be very big initially to the team or institution or country generating and managing and storing the data. But then the diversity of people and scientists and teams and medical doctors can exploit the data with minimal cost. And then analyzing those data is also related to optimizing resources, what I call here data ecology. Basically, these data had a cost, they are stored, they are costing to the community, so why not trying to learn from them as much as we can. However, important downsides are that they are not always adapted to the question that we may ask. They contain very heterogeneous information and they don't allow to control and estimate the biases, especially those related to initial steps in the generation of the data. So here I want to give you two examples of big data in the real life conditions or already available data, and illustrate the impact that biases can have on the conclusions and interpretations of the data. And first I will illustrate with one example biases that can occur at the level of the computational analysis and interpretation. And this is, to me, a very striking example of how different ways to analyze data can lead to opposite conclusions of an article. So the first paper shown here, published in PNAS in 2013, is from American consortium that actually generated a lot of data to address different questions in human severe injury, different or inflammation, and how the host response and host inflammation basically responded to these different types of stresses and injury. And in parallel, they have studied a diversity of mouse models for the same types of injury. And having generated tons of data, especially transcriptomics data, they asked whether the changes they have seen in the human disease parallel the changes observed in mouse models. And they found that it's not the case. And basically that there is a very, very low correlation, sometimes 0.1 or even less, in comparing gene changes in humans versus mouse models. And very surprisingly, 2 years later, a manuscript published in the same journal has an opposite conclusion. So from poorly mimic, they go to greatly mimic human inflammatory diseases. They analyze the same data, but using a different strategy and focusing only on the genes that change both in human and mouse, and then trying to correlate only those genes. In that case, they find a much greater correlation, and hence this conclusion of their work. So for those interested, I let you decide by yourself, you know, which one is the conclusion that you feel is more reliable to what actually happens and to the biology. But this was really to show how a bias, because this is a bias in a way, it can be a positive or negative bias, but this is a bias in the analysis that can dramatically change from one extreme to the other, the conclusion of an article. And a second example that concerns biases that can occur at earlier stages, the ones that are not very easy to track when we exploit already available big data. And this example is taken from the topic of our webinar, which is COVID-19 disease. And this is the Lancet paper analyzing the effects of hydroxychloroquine or chloroquine with or without a macrolide in the treatment of COVID-19, that you may have heard of because it has been discussed a lot and has generated a lot of controversies. It was published end of May in the Lancet and retracted a few weeks after. So why that? What were some problems in this study? Well, first you see the ambition of the study is huge. Basically analyzing 98,262 patient files. So it's a huge number, which goes to, okay, maybe we will learn a lot from those patient files, but there will be difficulties in addressing biases, heterogeneity in the patient populations and records. So in the end, about 96,000 patients were included in the study, 14,888 in treatment groups, you can see here the 4 treatment groups, and 81,000 in the control group. The control group is in fact all the other patients that have not received chloroquine or hydroxychloroquine. So you can see here the huge imbalance between those groups. And the fact that, as in any of those retrospective big data type of studies, basically we don't control the composition of the groups. So the control group is called control, but in fact is not controlled at all. It's very, very heterogeneous, and it would be very difficult then to compare the groups and lead to reliable conclusions. And so here I pointed to 4 possible biases that I think are worth mentioning. It's not the only limitations of this study. So I told you about the heterogeneous and ill-defined control population. Then there may be baseline differences in severity between treatment groups that would bias the outcome of the patients in terms of survival and morbidity. And why that? Because we may imagine that medical doctors would be more prone to give chloroquine or hydroxychloroquine, which are not approved for this type of viral infection, in patients that are more severe and in which they feel that they need to do something, otherwise they may be at high risk of mortality and severe morbidity. Then in the way they evaluated severity, there is also possibly a bias because they use this QSOFA score, which is a simplified severity score that actually was used because the data to generate the score are limited to 3 very basic clinical variables, and that's the ones that were available in the patient's clinical files. But it didn't allow probably sufficient granularity to differentiate between the initial severity, and this also may have biased the outcome of some of these groups. And then because of the lack of visibility and control over the early steps of data generation, even if here it's clinical data, there has been also limited QC and also very difficult to perform quality controls on the data. And that's one of the reasons that led to the retraction, because the authors, when they asked having access to all the raw data from the data providing company, basically they couldn't have access, and they said, okay, we retract the paper because we cannot put sufficient confidence in the conclusions of that paper. So really a big difficulty to analyze, identify and control this huge number of possible biases when analyzing a big volume of data for which we don't control the data life cycle. So on the other, let's say extreme, other type of data that we can use in this big data research are coming from controlled clinical and biomedical research, and in those cases we would have the visibility of all these steps. So it leads to different types of questions, different challenges, and again the pros and cons. So the positive aspects of these types of controlled experiments, a clinical trial being an experiment by itself at the level of a patient population. The data sets are then better adapted to the question that we're asking. We can control over biases and estimate the nature and impact of persistent biases that cannot be completely controlled. The negative sides are the cost. We need to generate everything from scratch. We can only generate a limited number, a limited volume of data and of data types related to our resources and to the ambition of the study. But we certainly, an individual team cannot match the volume of data that can be generated by an institution or a whole country or a huge consortium. And there is a time to generate the data because all these studies are prospectively performed. And so two examples, in the context of COVID-19. This is a study from the group of Genomic in Israel, having addressed a specific hypothesis using big data in the form of single-cell RNA-seq, in which they have generated a method to identify viral sequences in individual cells. They have classified individual cells as infected or bystander, and they could analyze separately the transcriptome of individual cells and relate it to the presence or not of viral sequences. And this was very informative on that specific question. There are so many questions that cannot be addressed, but you see here the potential of controlling and streamlining the process towards a specific biological question. And a second example from the group of Catherine Bleach at Stanford, which addresses a different type of study, an atlas study, so systematic characterization of a system, and in that case is a single-cell RNA sequencing of peripheral blood mononuclear cells in severe COVID-19. And you see an example of the different cell populations that were identified, and of course a lot of work has been done on the biological pathways expressed in those cells. And again, a more limited study on fewer patients, but the ability to control the quality of the data, the potential biases, and some of them are discussed in the article related to the treatments that the patients have received, for example. And so a better visibility on what can be concluded and what maybe cannot be concluded, or at least with sufficient level of certainty. And I show you an example from work that we are doing in a European consortium that is called Immunade, is this attempt to basically control all this data production line from a big patient population suffering from autoinflammatory diseases. In a European cohort, we plan to recruit over 700 of patients and generate multi-omics data, still keeping the control over the whole process with very detailed standard operating procedures, the centralized data management and data analysis that hopefully will be able to account for potential biases, because we know there will be biases in our study, but the fact that basically we set up everything from scratch allows us to identify potential biases and to correct them, and if not corrected, at least to account for those biases in our conclusions and interpretation of the results, in order to avoid examples such as I previously presented, meaning conclusions that would not be robust enough. So I would like to end with this slide, presenting the two different types of data that we can work on in terms of big data in the context of disease epidemics, such as now the COVID-19 disease. But of course, these strategies can be applied to any disease setting. And on the left, you have the real life or real world data or already available big data for which we don't control the data generation process, but that can still be a very important source of information at a very lower cost for the teams that accessing those data. And on the right side, the controlled data sets, biological or clinical study, that enable a much better visibility and control over biases and integrating the biases in the analysis and interpretation, but that are costly, usually more focused, and taking time to generate. And these approaches are not at all exclusive to each other, and ideally they should be both exploited by individual teams or institutions or international consortia, but having in mind the strengths but also the limitations of each one of those approaches, so as to learn as much as we can from them without falling into inappropriate or weak conclusions related to these very large number of possible biases that take a lot of difficulty to identify. Thank you very much, and thanks again to FAI²R for setting up and hosting these webinars. Thank you.